Hi and welcome to the 1.3 membrane structure video for IBDP biology. Here we've got the section from the IB guide. It's worth noting the application and skills section, as this tells you what you actually need to be able to do as opposed to the understandings, which is what you need to know. Here is a eukaryotic cell, and the plasma membrane is illustrated in blue here around the outside of the cell. Now the plasma membrane is made up of two layers of phospholipids, and therefore it's known as the phospholipid bilayer. You can see two layers just here in this diagram. Phospholipids have hydrophilic heads and hydrophobic tails. Now because they have both hydrophilic and hydrophobic areas, we can say that they are therefore amphipathic. Here you've got your hydrophilic head made up of phosphate and glycerol, and then fatty acid tails, which are hydrophobic. You don't need to know all of this specific structure in here. The plasma membrane also has proteins and cholesterol embedded within it, and sometimes it's referred to as a fluid mosaic model, and that's because all of these proteins shown here in blue that are embedded kind of make it look like a mosaic if you look at it from the top here. So if you look at just this section here, you've got all of the yellow phospholipid heads and then these embedded proteins which make it look like a mosaic. And it's called the fluid mosaic model because you have cholesterol within the membrane, and the cholesterol within the membrane regulates fluidity, so the membrane is said to be fluid. Here's a more detailed diagram of the membrane. So we have here a sugar side chain. Now the sugar side chain here is attached to a protein shown in blue, which makes this whole molecule here a glycoprotein. You've also got the phospholipids shown here, so you've got the hydrophilic heads in pink and the hydrophobic fatty acid tails in blue. You can see that the heads are on the outside here, so this is the outside of the cell, and the heads here on the inside of the cell. Now on the outside we would have extracellular fluid, on the inside you would have the cytoplasm. Both of those are water based, so that's why the heads are on the outside, so they are touching the water because they're hydrophilic or water loving. And then you have the tails on the inside, which are hydrophobic. Did I say hydrophobic for these? The heads are hydrophilic, water loving, and the tails are hydrophobic, water hating. So they point inwards. And actually, if you took lots of separate phospholipids and put them all into a beaker of water, they would actually form a bilayer because these tails don't want to be touching the water. You can also see that the heads are polar and the tails are non-polar. We also have proteins embedded within the membrane. You have integral proteins that span the membrane, so this goes all the way through the membrane. And then you have peripheral proteins which don't span the membrane, so this protein doesn't go all the way through, it's just here on the inside layer. Some obviously would be only on the outside layer. The membrane proteins have many functions. Now this could crop up as a short answer question or a long answer question within the exam, so it's worth knowing. One of the functions is that they are hormone binding sites, which is self-explanatory. Obviously just sites for hormones to bind to, and then obviously there would be an effect within the cell as a result of that. Some of the proteins are enzymes, so they catalyze reactions. Some of the proteins are channels, so you can see here that this protein is a channel, so it's making a kind of tunnel through the membrane for things to move in and out. In topic 1.4 we'll learn more about that. You have some of them being pumps, so pumps are used for active transport. You should have some knowledge of active transport from some prior learning. Now the pumps require energy in order to work, so that's what makes them different to the channels. Channels don't require energy, the pumps do. The proteins can also have an adhesive function to allow the cells to stick to each other. You've also got cholesterol in the membrane. Now the cholesterol regulates fluidity, and the more cholesterol there is, the more rigid the membrane, and the less cholesterol, the more fluid the membrane. Now, if you imagine a football being pumped full of air and being quite hard, that would represent a membrane that had lots of cholesterol in it, so it was rigid. And if you imagine a football with half of the air taken out of it, then that would represent a membrane with less cholesterol in, or a more fluid membrane, so it can move around more. So when we talk about the membrane being fluid, we don't mean that it's a liquid, we mean that it can move. We've also here got a timeline of the development of the theory of the membrane model. Now, initially, Gorter and Grendel suggested that the plasma membrane was a bilayer, and we do now agree that the membrane is a bilayer, but they were the first people to suggest that. That was in the 20s. In the 30s, Davson and Danieli proposed that there were protein layers on either side of the membrane, so they suggested it was a kind of sandwich. So it had a protein layer, then it had your bilayer, and then another protein layer. Now we know, or Singer and Nicholson proposed that the proteins weren't in layers. So although Davson and Danieli were the first scientists to suggest that there were proteins, they got the location of the proteins wrong. So Singer and Nicholson suggested that the proteins were integral and peripheral and embedded within the membrane, as opposed to being a layer on either side of the membrane. This is the theory that we accept today, that we assume is the actual structure of the cell membrane, but there was work done in order to disprove this theory of the membrane, the Davson-Danieli theory, and there were a few things that occurred that allowed us to disprove that theory. So number one was freeze-etched electron micrographs. Now this is where we freeze the membrane and then break it and look at the cracks that appear when you break the membrane. So because the proteins were embedded within it, the cracks cracked where the proteins were, so the arrangement of the cracks within that membrane suggested that actually the proteins were embedded as opposed to layers. There's also been development in techniques, lab techniques, which allows scientists to look more closely at the proteins, and they now know that the proteins are of different sizes, which suggests that they're not in layers, they are actually embedded. And then finally, you can tag the proteins in the membrane and look at whether they can move around in the membrane or whether they don't move around. So if they were in layers, you would assume that the proteins wouldn't move around. Whereas because they do move around, we can assume that they are actually embedded within the membrane. And that ends 1.3 membrane structure.